Hello, everyone, and welcome to our special KNI Distinguished Webinar. And today, we're super lucky to have Dr. Ellis Meng, who is a professor of biomedical and electrical and computer engineering at the Viterbi School of Engineering at USC, little school down the road, right there, where she also serves as the Vice Dean of Technology, Innovation, and Entrepreneurship. I would like to point out that Ellis is a lifer. She received her bachelor's degree in engineering and applied science, master's degree and PhD degrees in electrical engineering from Caltech in 1997, 98, and 2003. So clearly we were doing something right in treating her well for her to have stayed with us for such a long time. So we're delighted to host Dr. Meng here today. Her research interests include biomedical microelectromechanical systems, biomems for short, implantable biomedical microdevices, and multimodality integrated microsystems. So you can probably already hear the connection with electrical engineering, with medical engineering, with mechanical engineering, bioengineering, and every single discipline that we have here at Caltech. Today, Ellis will present her team's latest progress on polymer-based microfabricated medical implants, from invasive polymer interfaces to nervous tissue and to electrochemical sensor systems for treating hydrocephalus. Before we get started, I'd just like to remind all of you to please submit your questions through the Q&A feature. We will definitely go through them. I will read them at the end of the talk. And hopefully we'll have Dr. Meng for a little while until 5 to be able to answer all the questions that come up. This seminar is being recorded and we will post it on the KNI's YouTube page in case you need to leave early or if you're not able to join us today. Now it's my pleasure to welcome Ellis. Thank you so much for being with us today, and we're looking forward to your seminar.
Well, thanks so much, Julia, for the introduction. It is good to be back, even though this is a completely virtual setting, but hope that this will be the first of maybe future interactions to come where we can actually group again in person. So I'm going to talk a little bit about some of the stuff that we do at USC in my research lab. Some of the work that I'm going to touch upon today will be within the sphere of one of the startup companies that have originated from my lab, Sincere LLC. And so I want to get that disclaimer out of the way up front. So since, I'm not sure where your backgrounds are, I'm going to start off very basic and then kind of work our way up through more in-depth topics. And so to start off with, I want to give kind of the motivation of building systems using microfabrication technologies for both medicine and science. And I think that the two driving reasons are that, with these tools, I can actually help colleagues that actually are in the sciences ask questions that they couldn't ask otherwise. And in the medicine also come up with therapeutics, diagnostics, monitoring technologies that wouldn't exist without these technologies. And the underlying theme, or one of the things that ties all of my research together, is we utilize tools that originally developed in the microfabrication world for building integrated circuits, things like transistors and so forth. And instead, we adapt them to work in implants, specifically in my lab. And we do a lot of work that uses MEMS tools, so microelectromechanical systems. Those of you who've heard from YC, my graduate advisor probably have heard this word over and over again. And I think that for those of you who don't know what it is, I think the characteristic feature is that they have some small component, and that small component or feature could be anywhere from nanometer to millimeter. And when we start to put together the different structures, we start to build systems. Systems could actually get much larger, but the active element usually is much smaller than that. And so the reason for investigating these technologies is that they really expand what we can do. When I was at Caltech as a graduate student, I was in electrical engineering, but what I was actually doing was pushing tiny amounts of fluid around using devices that were microfabricated instead of pushing electrons around. And so we could start to interact with the world in a different way. And there are now many commercial devices out there that utilize these that we use every day without even thinking about that. So the underlying technology that we use in my lab is actually quite simple and involves a series of steps, a recipe, so to speak, that allows us to construct devices that are then used for a variety of different applications. And I'm going to start off with just orienting the group on what these devices look like in case you're unfamiliar with how this is done. So In general, in my world, we speak a lot in cross sections. So for many of you, this could be quite familiar. And in the center is a sequential set of steps that represent what we do to actually get the device that we want at the end of the day. We work in thin materials. In my world, largely one polymer, our favorite polymer is perylene. This is a trick that I learned at Caltech. And then we use as our active element, so to speak, a metal, platinum. So very simple. We have these perylene, platinum, perylene sandwiches that we develop using a flat substrate. Our favorite flat substrate is silicon as a carrier. And then we later on, after we're done, peel these off, and then we end up with these flexible medical implants. And on the right are some images that I'll explain in more detail as we move on, but they're examples of devices that we make in the lab. We have done a range of different projects over the years. Currently, my lab is largely focusing on neural interfaces, and I'll talk a little bit about the work we do there. And we also more broadly work on sensors for different applications. And I'll share one specific application that we've been working on that's getting closer to the clinic. There's a number of other things that we do, developing some of the underlying technologies to allow us to be able to build up more complex systems. And I've also done some work in drug delivery earlier on, but I'm not going to talk about today in the interest of time. So to start off, I'm going to give a little motivation for wanting to use microfabricated systems for studying the nervous system. And in particular, one area of great interest is how do we better understand the brain? And I think that much of the progress in neuroscience is enabled by the fact that we have tools that are increasingly more sophisticated and that allow us to interact with nervous tissue at different scales. And so the brain, it turns out, is very difficult to interact with because it does span so many different length scales. And so this kind of gives you in one slide an idea of the complexity that we're dealing with. And with microfabricated tools, we're not able to completely get down to the level of molecules, but we can talk at the level of neurons. So neurons on the order of 20 microns or so, this is very easily achievable in terms of building an electrode-based interface to neurons using microfabrication technologies. And that's much of what I'm going to be focused on today in the context of neural interfaces. And so there are, I want to acknowledge a number of other technologies that can be used to interface with the brain. I'm going to focus largely on the electrical modality for a variety of reasons. There are optical technologies out there today, as well as other chemical ones. But my lab is largely done is try to develop different technologies to interface with not only the brain, but other parts of the nervous system that have a very different anatomy. Let's start up our conversation with the brain. So it turns out that what many neuroscientists want is a great deal of information that can only be attained at the single cell level. And so it is possible to get information at with different levels of invasiveness. You can record from the outside of the body, but to really get at that single cell information, you really have to go with an invasive approach. And the best way that we currently know of doing this using electrical modalities is by establishing a one-to-one interface with a cell and achieve something known as an intracellular recording. So this is not a scalable technology. So this is depicted right here. I don't know if you can see my mouse. It's not a scalable technology and does require that you search out and find a neuron. And so this is not something that you can implant and do at scale over thousands of neurons, so to speak. But it is possible to move a little bit further away from the cell and be adjacent to it, say within 50 to 100 microns or so, and still get good information. When you're at that level, you now run into something called an extracellular potential. You can record these for single neurons or for groups of neurons. And that's really what we're trying to achieve with the microfabricated interfaces that we build in my lab. So progress in this field has actually significantly improved over the last several decades. In the early days, much of the recording was done using very simple, small diameter metallic microwires that are insulated all along the length of the wire, but de-insulated only at the tip. So you have a very small, well-defined region that can make an interface with the nervous tissue. Later on, as silicon technologies developed, this was then slowly replaced with, although they're still used today, the microwires, I should say, but slowly replaced in a lot of experimental labs that wanted greater scale by silicon-based devices. And so in silicon, if you look at the Utah array down here, you can mimic the geometry of and the layout of the microwire devices. You have here a series of different tines that are de-insulated only at the tip and insulated all along the shank. This is a so-called bed of needles approach that can be inserted in the brain and interface, actually not just the brain, but the peripheral nervous system as well. So this is not super space efficient in the sense that you only have a single recording site at the very tip, and you're in some sense wasting the volume that's along the length. And so another group at the same time realized that you could take these microfabrication technologies and put electrodes all along the length of these tines. And then out of that came the so-called Michigan approach. So we have the Utah array that came out of University of Utah, and sort of that name is kind of stuck, although commercially it's available through a vendor now with a different name. And then we have the Michigan arrays that kind of look like this, and you can see in the image, hopefully, the different electrode sites all along the shank that can be packed in. And so you can access a greater volume in this way, and you can access cells all along the length as you put this device into the brain. So with these advances, we can certainly increase the density. However, scaling of connections external to the body that allows to connect to the electrodes that are in the brain or in other parts of the nervous system is actually quite complex. And so there's a growing realization that to address the scaling problem, you have to make use of microelectronics. And so in the bottom right-hand corner here, one of the benefits of using silicon is that in the same material on the back end, typically, you can start to pack in some electronics and reduce the amount of wires coming out of this device. And I'll talk a little bit more about the scaling problem later. But in general, what you notice is that over a period of time, you get roughly every 7 years, a doubling of the number of neurons you can record from, drawing a Moore's law-like analogy between these technologies and microelectronics. So I mentioned earlier that one of the motivations for going to more electrodes is that neuroscientists want more information. There's a lot of information we can extract, and even if we're talking about thousands of electrodes, we still sort of pale in comparison to the number of neurons within the brain. But baby steps, one thing at a time. So in addition to that sort of scale problem, there are also many other requirements that we might want to impose upon our system. We might want to record from many different regions in order to dissect different circuits and understand brain function. We may want to record from single neurons and large groups of neurons. We may want to understand different phenomena as they occur on different timescales, from milliseconds per years all the way to years. And then when we make these interfaces, we want to implement them in such a way that we minimize the damage that occurs within the tissue. And so there are many challenges. Some are very much engineering challenges, including how do we increase the density of these devices. There are also surgical challenges. How do we design devices such that when we do surgical implementation, they can get to deep targets? A lot of what I showed you earlier involve devices that can only reach some of the more shallow targets, not deep targets deep within the brain, so more than a few millimeters. A big issue is how do we maintain the quality of recordings over time? This turns out to be quite a difficult problem that many people have tried to address from different angles. I'll talk about that a little bit in our approach to that. And then one thing that is not necessarily very interesting from a scientific perspective, but is very interesting, very essential for building devices that actually work in the lab in the long term, is that we have to consider what it means in terms of packaging. So we have an interface. What does the rest of that device, that system look like that allows the neuroscientists to interface with that and get the data that they want over the long term? And so I'll also address a little bit of that as we move on. So I mentioned earlier that longevity of the interface is a problem. And what I mean by that, to be specific, is that when you implant a device, typically you can get decent recordings for a short period of time. Over time, the presence of the implant will cause a scar to form, as well as a number of different activities that are undesirable. They all constitute an immune response. And what that can do is that, if you look at this cartoon down here, you want to have neurons in close proximity to the electrode. If a scar forms, or if there is just damage to neurons, you can cause neuronal death, or you can cause neurons to be pushed away from the electrodes. And so that can contribute to a degradation of recording quality over time. This is a very simplified view of what's actually going on. There are many complex processes at play. But one thing that we are beginning to see more and more in the evidence in the literature is that by moving from very stiff materials that form the backbone of the probe that is placed into the nervous tissue to softer, more compliant ones, in general, you produce less of a severe immune response and can have a more stable device tissue interface over a longer period of time. And so this cartoon here and then the evidence down below, we're looking at staining of in the bottom comparison between stiff and compliant kind of demonstrates that indeed there is some effect by moving towards softer, more compliant materials. So here's a chart that gives you a sense of what we can do as far as improvement by changing the backbone from something like a stiff metal or silicon to a polymer. We gain about a factor of 100 if we're moving to a polyimide or parylene. We can do better by going to silicone, but silicone is not necessarily a great engineering material to work with at times to build certain types of devices. And so most of the field is kind of stuck with polyimide and parylene at this time. If you look on the bottom, these numbers here are Young's modulus. And so as you get to softer and softer tissues like spinal cord and brain, you'll notice that even if you can reduce the stiffness of the backbone that's used to construct the device, you're still many orders of magnitude off. But still, the softer mechanical backbone seems to help. And so the solution isn't perfect, but is a step in the right direction, we hope. And so the approach that we've adopted, and I've shown you a little bit of this earlier, is to take a very simple polymer metal polymer construction and build Michigan-style like devices that interface with the brain. And so here's a summary of the materials we use. I mentioned this very briefly earlier. We fabricate all of our devices on a silicon substrate. We use this because it's conveniently available. It comes very flat in a number of different sizes. And then we use Parylene C, which is a transparent polymer, as both the building material as well as the electrical insulator. And then platinum is the conductor that creates the sites for recording from different neurons, as well as allows us to make connections externally to the devices that are implanted. And the reason for choosing these materials, it's sort of a safe approach, and that ultimately the goal is to be able to develop implants that can be used clinically. And so we've chosen to use materials that are present in other FDA-approved implants as a lower risk approach to attaining that one day. The construction, here's a cutaway view, a cross-sectional view of what a typical device might look like along an electrode site as well as along a trace. We use typically 5 to 10 microns of parylene per layer, and the electrodes are about 200 nanometers. So that gives you an idea of scale, and the width and the specific geometry will vary based on the application. And I'll kind of show you what we can do with these devices. So using this very straightforward method or structure, what we can do is build a wide variety of different kinds of microelectrode-based interfaces to different parts of the nervous system. And so the simplest ones to implement in theory are what we call surface electrodes. They sit on top of nervous tissue. And so the image here of the device on the top is a device that's intended to sit on the retina. You're looking at an as fabricated device. The actual electrode interface is the leftmost tip here. You'll see a series of 8 different electrodes, of which there are 4 different sizes. This is an experimental array because we don't know the most appropriate size. This is intended for use for stimulation of the retina and specifically for use in the rat eye. The rat eye, the rat, I should say, as an animal model is chosen because it's very convenient to do visual experiments in. However, the size of the rat eye makes it very difficult to access the retina. And if you look at a cutaway view of the rat eye, the internal anatomy is a little bit different in the sense that the lens occupies a huge amount of space within the volume of the eye. And so that leaves us very little space to work with to be able to introduce an array onto the surface of the retina and imposes a lot of constraints on what the device needs to look like to get in that space. And so this initially flat array is formed using a thermal process and taking advantage of the thermoplastic nature of the purlin to have a very tight bend where the array enters into the eye, and then a more slight bend to allow the array to sit with the electrodes facing the retina. And so this is work that's being done in collaboration with Jim Weiland's group at the University of Michigan. I should mention before I forget that in my lab, we only build devices and we don't do any animal work. So anytime you see animal work, this is work done in collaboration with different groups around the US. So on the bottom is a slightly different device. It's also a surface array, but it's intended to interface with the peripheral nerve. And so the peripheral nerve anatomy is quite different from the retina and also quite different from that of the brain. And typically the processes that you want to record from are hidden behind protective tissue, the epineurium and perineurium that surround those nerve bundles. And so what happens is that although this cuff where you wrap electrodes around a nerve is minimally invasive, it doesn't give very good signal to noise ratio. You don't get a lot of selectivity as a result. And so we want to preserve this kind of minimally invasive interface without having to plunge electrodes in to get closer to those fibers for recording. And so here the approach was actually to integrate some microfluidics into the electrode devices. And the purpose of this is actually twofold. One, we want to infuse at very focal sites on the nerve collagenase, a lysing agent, to actually remove some of that, the collagen content and the protective coating, and then subsequently follow that up with another dose of a growth factor. And that will allow, we hope, is for processes that are exposed to grow out into the channels within our device and get closer to the embedded electrodes contained there. So this overall device was designed for that purpose. In this image here, you can see colored in blue. These channels are filled with blue dye, just to allow for visualization. The nerve goes here. And so the rest of this device is really for securing the cuff to the nerve and for packaging. So we have areas where we can allow microfluidic access to the device as well as electrical access to the different electrodes. Another format we can use are penetrating devices. And this is kind of where I started the introduction, devices that penetrate into the brain or into other nervous tissue to get the electrodes to the target site where you want to record from or you want to stimulate from. And so this is a device that we did several years ago that has a very kind of a fancy structure and was intended to be used in rat motor cortex. And so these were used along in the rat and designed to look at whisker movements. And so they didn't have to go very deep. They went about 2 millimeters down. These are arrays that have been built and packaged into this housing back here. Each device contains 4 electrodes. Each probe contains 4 electrodes. And on the inside and 4 electrodes on the outside. And what I mean by that is there's this conical shape. There are actually 4 electrodes that are maybe a little bit hard to see on the inside of the cone, as well as 4 electrodes on the perimeter. This specific device was actually coated in Matrigel, which was then soaked in growth factor or also anti-inflammatories as we were trying to look at how to promote tissue integration with the device in the cortex. So that's one example. And another example that's a bit simpler, but difficult because we're trying to reach deeper brain targets is the one shown on the right-hand side. So this device is intended to interface with the hippocampus and different regions of the hippocampus that are involved in memory formation. And so you can kind of see that these pastel overlays are taken from a rat brain atlas. And these electrodes are actually placed specifically to match the anatomy that we pull from the atlas. And so we call these conformal electrode arrays. Here we have 8 tines, each with 8 electrodes for a total of 64 on this device. And I'm showing you the tip as well as the back end where we actually connect to external recording systems. If you have one of these very long floppy devices, it turns out it's difficult to implant that into the brain. You have to support these soft polymers. Otherwise, they undergo mechanical buckling and essentially don't insert into the brain when they're not stiff enough to actually penetrate. And so the mechanical trick that we play is we temporarily shorten the length of the probes, as shown in this middle set of figures here, by embedding the back end in a biodissolvable material called polyethylene glycol. And so what we can do then is leave the exposed length quite short, and this becomes much stiffer in this format, and we're able to insert the probes bare into the brain. So we don't increase the cross-section to insert them into the brain. And this is in contrast to the prior approach on the left-hand side over here, where we're using insertion tools that actually do add a bit of bulk to the system and do more damage. So with both of these, I want to stress that we're able to get long-term recordings. And so I took a snapshot of data that we took, roughly a year out for both of these devices. And we're able to get stable long-term recordings in the brain with both of them. And so that's a significant improvement over some of the silicon-based devices where you can get weeks, maybe a couple of months, but it's difficult to have great longevity of electrode sites over years. All right, so the previous devices I showed are relatively low channel count. And so, at 32 and 64, it's really not at the level that a lot of neuroscientists would like to have. It's possible to scale to hundreds of sites with some of the traditional technologies I mentioned earlier like microwire. But if you look on the back end, the amount of packaging to connect to each one of these is pretty substantial, and that becomes experimentally difficult to implement, especially on small animal. It's quite bulky. These are quite prone to failure. In many cases, people will report that the connector has snapped off, leaving a device in the tissue, but essentially it's rendered, the experiment has to end because you can't connect to any of the electrodes that are remaining in the brain. And so, the scaling related to packaging becomes a bigger issue as you go to higher and higher channel count. This is also true of this Utah electrode approach, where you can kind of see a very thick, rigid wire bundle coming out of the back end and connecting to this very large connector. Both of these approaches on the left-hand side could take advantage of silicon microelectronics to be able to reduce the number of wires coming out. And this is by using just simple multiplexing. So sharing some lines and then just reducing the number of wires that come out. And this is very easy to do in the Michigan approach, where you can integrate electronics on the back end of a probe. And they've explored one modality, and there are many other groups that have actually done this as well, using slightly different approaches. And in general, the message is that you could start to think about devices that are at thousands of sites or beyond. And there are devices available commercially now that will allow you to get close to about 1,000 sites. Right? So this is all positive progress. But with these silicon devices, longevity, as I mentioned earlier, remains an issue. Right? And so we want to be able to do this in polymer-based devices, but we don't have the benefit that the silicon happens to be there. And so our approach is actually to take the silicon and bring it to the polymer electrode array. And so this kind of captures the overall approach. If we increase the number of electrode sites, which we can easily do using microfabrication, nanofabrication, and then add on the silicon electronics into the back end of the probe, we can greatly reduce the number of wires and complexity of that packaging that has to come out of the brain or out of the body, and start to realize systems that now have thousands of recording sites. There are other approaches out there I want to acknowledge that also look at the scaling. This is sort of our take on that. And so over the last couple of years, we've been looking at this from different angles. We've been looking at improving the microfabrication process to allow us to build higher density arrays, and also taking full advantage of both the front and back side of the devices. And the devices I showed earlier, all of the electrodes sit on one side. We're also looking at ways in which to integrate application-specific integrated circuits into the devices to deal with both signal processing and multiplexing, and then investigating what is the impact of actually scaling the number of electrodes, or not electrodes, but number of shanks that go within the brain and the impact on the tissue. We don't want to display so much that there's actually nothing left to record from. All right, so very briefly, I'm just going to show you some of our more recent results of devices that we fabricated and are in the process of testing. So these are our newer devices. We have both a hippocampal design and a cortical design. As I mentioned earlier, the major difference here is that we now have electrodes on both the front and back side, so 32 sites on each side. And so if you have 8 of these, there's 512 electrodes on each one of these arrays. Now, we are still limited until the application-specific integrated circuits are developed to conventional connectors. And so you'll see on the back end, the wiring out is not very dense. And that's because we're just using standard 64-channel connectors to be able to address a subset of the electrodes in our current devices to do some initial testing. In order to get the electronics on the back end, we looked at many different approaches all at the same time. And these are the approaches that we looked at. We looked at using conductive epoxy, making rivet bonds using ultrasonic wire bonding, purchasing anisotropic conductive film, and establishing many connections all at once using heat and pressure. And then we developed our own method, which I liken to creating waffles in a way, called polymer ultrasonic on bump. And here what we're doing is we're taking the ASIC chip represented by the gray down below. It has some connector that we want to make a external connection to. We put a coin bump on top and then invert one of our arrays, and then take a tape automated bonding tool, which actually does kind of look like a waffle, miniature waffle iron. And we press down and we're able to make very tight pitch bonds in this way. So part of the reason for looking at 4 approaches at once is that we weren't sure exactly which one would pan out. And it turns out all of them work. But for a variety of reasons, we actually chose to proceed using the very bottom approach, polymer ultrasonic on bump. And in the interest of time, I won't go into all the reasons why. And so in doing this, so here's some other views of those tab tool enabled bonds using the pub approach. We were able to bond to a custom ASIC that was made by Hossein Hashemi's lab. Hossein is also a Caltech graduate from roughly the same time that I finished up. And so this is a chip here in the middle that one of his graduate students produced. It's a single channel programmable stimulator. And this just shows you that there's 30 odd bonds that we created here, all very tight pitch. And this is a zoom in view of just what the interface looks like. But exterior to that, the chip is down here. There are a bunch of wires that we have to hook up that are still very large. This is just for testing purposes to be able to talk to the device and do some initial testing. And then we're also using a commercial device created by Intan in a ball grid array package. And we can also use this bonding to attach to that. So this is a very versatile technique that allows us to adapt to different electronic circuits, sort of regardless of their format, connection format. On the bottom here are just some results that we got recently, where we took a simple metallic microwire, bonded it to the chip, to the stimulator output, and put that into a saline bath, and then recorded the output of that using another tool. And so this and we've been able to program in different stimulus pulses. This just confirms we're able to read out using this device that's been concatenated, a system that's been concatenated together from different components. So I've kind of shown you the direction we're headed, trying to deal with a lot of the scaling issues as we increase the number of electrodes. We are doing a lot of work in developing ways to automate stacking, waiting for the next ASIC to come out so that we can do the integration with the multiplexing, as well as ongoing in vivo studies. In the future, one of the areas we're interested in is even probing deeper structures, going beyond, say, 10 millimeters, which is kind of the the barrier right now. It's very difficult to make probes that are much longer because of the surgical implantation problems that I've mentioned earlier. It's hard to find a way to introduce them without creating a lot of damage. And this is important so that we can actually start to address even just the same structure of the hippocampus in different species. It's at different depths. And the image on the right kind of captures what it looks like as we go from rat to monkey to human. I mentioned that we're trying to do some stimulation. Most of the work I showed you is actually for recording. So we're tailoring electrodes for stimulation in collaboration with others, building new electrode designs. And then I italicized this. One of the things that's happened along the way is that many colleagues have asked if they can have devices. And so I've gotten enough requests that I've taken this sharing devices and dissemination very seriously, and this has really been enabled by new programs through the BRAIN Initiative. And so actually in fall of 2019, we received funding through the National Institutes of Health as a technology resource grant to open up what we call the Polymer Implantable Electrode Foundry, or PI Foundry. And this is a resource that is governed by a steering committee. It's run by multiple staff. It's a collaboration with my longtime animal collaborator, Dong Song, also at USC, and offers essentially free resources, free services as well, and training for researchers in neuroscience, for academic, I should say. And offers a range of different things. We have developed a standard probe. I'll show you an image of that in the next slide. We have the ability to do custom projects for different users, as well as build devices that users submit that they've designed. We also had intended to do a lot of on-site training in person to show how to do the implantations and how to do the animal experiments. That was interrupted, of course, by the shutdown. Those were supposed to happen last March, and we've quickly adapted to going completely virtual as a result. And so we're hoping that by doing this, a lot of the research and polymer-based electrode devices that is currently limited only to research labs that develop these devices can now get into the hands of different researchers. And so here's our standard probe, and then the image on the bottom left is actually one of our first shipments to different users. And this is actually based upon the work I showed you earlier of the hippocampal probe. It has a reduced number of shanks, but otherwise same number of electrodes and the same length as before. We've developed our own sort of custom packaging shown here, and this is also put together using the pub technique that I showed earlier. Just to give an idea of some of the other things that we do, we're influenced a lot by the services in the integrated circuit world for allowing researchers to access those integrated circuit runs. And so we developed a process that allows different users to share space on runs that we do, and that's illustrated in the cartoon on the right-hand side. Users can submit their designs. We don't really ask too many questions, just make sure that they don't violate any basic design rules. And then pack them in and fabricate these devices and send them back. In an ideal world, typically our turnaround is about a month. Currently, the USC clean room is shut down and we're waiting for it to reopen. So when that opens up again, hopefully in the summer, we'll be able to meet those turnaround times again. To make use of the resource, in case you're interested, we do have a couple of upcoming webinars, and you're more than welcome to attend, and you can RSVP using the link below. The other way to interact with us is we found that in many cases, neuroscience researchers don't know how to design devices by themselves, and they want someone to help them along. And so to sort of get access to this resource, we actually don't select projects ourselves. Our steering committee does that. And so you submit a simple proposal to us. It's reviewed by that steering committee. And if they approve, we go ahead and we make that using the resources available through the Foundry. And so the only thing that we ask users do is that we don't handle the implantations or the animal experiments. And so that's on the users. So with that, I want to switch over and talk about my second topic in the last maybe 10 minutes or so. Also in the brain, there is a condition known as hydrocephalus. This is somewhat rare, but perhaps more common than one might think. It affects typically 1 in 2 in 1,000 births and can also be acquired through trauma, through cancer, or through an accident. And to understand hydrocephalus, it's essentially a plumbing problem in the brain. You're within the cavity known as, or within space known as the ventricles, you have production of cerebral spinal fluid that bathes the brain and spinal cord. It serves as a cushion. It also is a way to, bring in nutrients as well as get rid of waste. And so there has to be this balance between the production of this cerebral spinal fluid and its drainage under normal conditions. In patients that have hydrocephalus, this balance is disrupted. And so typically what happens is you have accumulation of this fluid, and that results in commonly enlarged ventricles. The way this is currently treated today is that you essentially insert a piece of tubing into the ventricles and drain excess fluid through a pressure regulated valve into another part of the body, typically the peritoneal space, as shown in the image down here. In the ventricle, there is a proximal catheter that has a series of holes in it, and that is what allows the fluid to be taken up and drained elsewhere. So this simple device was developed in the 40s and 50s and essentially remained unchanged over the last, basically since then, despite many efforts to improve it. And improvement is really needed because these shunts, while they save lives, they actually do fail at an extremely high rate. The predominant cause of this failure is actually blockage of the holes within the proximal catheter that I just showed you. And so patients that actually depend on these devices, it's not really a matter of, if, it's a matter of how many times over the course of their life will they actually have to get a revision or a replacement of this shunt. This failure usually shows up in a very non-specific way. A patient may come in and have symptoms like headache, nausea, very vague symptoms that could easily be attributed to something else like the common flu. And also this evades detection using standard clinical means. The shunt sizes are such that it's very difficult to use standard imaging methodologies to be able to detect that this failure is going to happen. And so several years ago, in conversation with pediatric neurosurgeons, they have experienced this problem over many decades and really want something to be done about it. And they asked us to think about creating a self-aware shunt that could report back to them and tell them, no, it's not just a standard headache. You do have to go in and intervene. The patient is about to have a failed shunt. So down the road, our goal is to enable a fully smart shunt that has sensors that can provide this information to clinicians. This has actually been proposed since the 80s, but sensor technology at the time really wasn't ready to basically meet the constraints of a smart shunt. But I think now with sensor technologies that are available today, we can really start to think about this more seriously. And there are some other efforts to develop this. So one of our intermediate goals, ultimately we want a fully implanted sensor module, but it takes a long time to develop these full systems. As sort of an initial step, what we are doing is testing these devices or testing sensors we've developed and something known as an external ventricular drain, which is shown on the right-hand side. So these are devices that essentially mimic a shunt used to treat hydrocephalus. The one difference is that they're used very temporarily for a max of about 2 weeks in patients typically that come in with trauma. And so with sort of head trauma, one of the features is oftentimes increased intracranial pressure. And one of these devices is inserted to relieve that. And then there's outflow of cerebral spinal fluid through this. The device itself is not very sophisticated. There is a bag attached to the output. And the bag itself is attached to a ruler-like device that allows you to set the height and then the drainage level. Okay, so here's very quickly what some of these sensors look like. We've actually integrated within the same flexible substrate. There is in fact a substrate back here. It's sitting on paneling, which is transparent, and so you can't see it very well. There are a number of different sensors that have been arranged to be so that they are active in the direction of flow. A sort of zoomed out version of the device is shown on the right-hand side. And in there, we have flow sensors, pressure sensors, and what we call a patency sensor. And the patency sensor is named that because we're trying to look at directly the obstruction, the level of this obstruction of the different ports within the proximal catheter. So in the interest of time, I'll just talk about one of the devices, the flow sensor. Looks like my animation is not working correctly. So in the flow sensor, I'll just tab through it. The approach that we take here is we actually use the cerebrospinal fluid as part of the sensing circuitry. So we have electrodes that are exposed to the fluid. And in this system, we have a double layer that forms. And this is the sort of equivalent Randall's circuit that describes what's going on at the electrolyte interface. What we do is we heat the fluid up, and the heat is carried by the flow towards a pair of open sensing electrodes. And if we look at these sensing electrodes at high enough frequency, we essentially bypass these the double layer capacitance. And so effectively, we're looking at just changes in the solution resistance. It turns out the solution itself, so this material is going to be human cerebrospinal fluid, is quite temperature sensitive, and it's more temperature sensitive than common resistance temperature detector materials like platinum, right? So it's listed down here. And so the idea is that as this heat is carried downstream by the flow, we can actually detect a decrease in the solution resistance by measuring the electrochemical impedance at high frequency, and that then correlates to the increase in temperature. And so with that, we actually ran some studies in these external ventricular drains at Children's Hospital of Los Angeles. This is only using the flow sensor and some of the initial studies. And so this is our setup. The way these experiments work is that we are dealing with trauma patients. And so we really can't plan ahead as to when these patients will arrive. And so, when they do, we usually get a call pretty late, and then have to rush over there and assist with implementation of these. And so these are all done. We have an IRB approved at the Children's Hospital Los Angeles, and we've done a couple of these. And so I'll show you very quickly, and then I'll wrap up so that there's some time for questions. This is some of our initial data taken with these devices. So I mentioned earlier that the clinical external ventricular drains are very simple devices. The way flow is monitored and recorded by nurses is that they actually take the collection bag, this item here in the center, and they weigh it periodically. And so this data here is that volume that is weighed. We've converted that into a flow rate, and we're comparing this to our sensor, which is running roughly at the same time. And so very loosely, we can say that we are capturing some of the outflow that is supposed to be happening. But the comparison is really not that great given the sort of low fidelity of data that we're getting from the clinical external ventricular drain. Also, you'll notice that some of the flow rate here went negative, and that's because these sensors were not properly temperature calibrated. And so we now have that circuitry in our system, and we're looking forward to doing the next study with this now both in animal and in patients down the road. So some of this has been interrupted, of course, by the pandemic. So I'm showing you data that was taken before all that happened. So with that, I'll just quickly summarize, showed you some of the work that we've been doing over the last several years in the area of neural interfaces. The thing I'm very excited about there is the possibility now that we can share some of the technologies that we've developed over the years with others. And we've been able to actually serve research groups around the world in the early stage of our foundry. And looking forward to seeing hopefully an uptick as labs begin to reopen. And then the last thing that I talked about was a little bit of our work moving towards smart shunts, which has been a goal since the 80s. And I think we're getting closer to that as we're able to miniaturize devices and have them work in the body for longer periods of time. So to wrap up, I want to make sure that I acknowledge all of the funding agencies that have supported the work. And then, as I mentioned, all the work you see involving patients or any animal work was not done by my lab, it's done by my collaborators. And so some of them are listed here. and then the various students that have contributed to the work over time, both current and past. And so with that, I'm happy to answer any questions. I think we have a few minutes remaining.
Thank you so much, Ellis. This is such a fascinating talk. I think that the translational nature of fundamental science is something that we could really learn at Caltech and really make learn a lot from you. And we have a whole new department here called medical engineering that emerged since you left. And in fact, YC Tai was instrumental in founding it. He's definitely one of the founding fathers. So we're so thank you so much for teaching us this and sharing some of your work with us. Okay, so everyone who is still online, please feel free to submit your questions through the chat box or Q&A. I see a Q&A box actually. I also have a few questions that came in before. So one of them is, can a heart pacemaker that responds to exercise level be made with these devices? So can you monitor in situ your heart rate or your level of activity as you're exercising?
I mean, you can certainly make anything. I think what drives us to pick up a project is oftentimes asking, is that really a good way of doing that, right? Especially for implantable devices. If it doesn't need to be invasive, if there's not a good reason for it, then, oftentimes I'll talk to different collaborators and recommend that they talk to somebody else or do something that is non-invasive. You can certainly adapt pacemakers and various things to monitor all sorts of parameters. In the case of exercise, I think you know what it is that you're after. One of the challenges, and I think this is maybe getting a little off topic, but is somewhat related. One of the goals is to build devices that are more closed loop, right? And so one of the challenges that we face, and I definitely faced this early on in drug delivery, is how do you know what it is that you're supposed to sense, right? Knowledge is not completely there. Medicine cannot offer us solutions on what biomarkers to detect. And so that remains a bit of an obstacle, a roadblock to developing, I think I would say, next generation closed loop devices. But certainly, I think, can you modify pacemaker? Yeah, you can do that.
All right. So I guess that's a good question because people are exercising. So I guess they're curious about it. It's good. Okay, here's the next one. In 10 years, how widespread will this technology be? Let me rephrase this question. If we were to look a decade ahead, do you think that these kinds of implantable devices and just wearable devices will be mainstream? And will they ever be mainstream, or will they always be in the niche applications?
I think to some degree, there's always going to be a nichiness to this. And that's partly because of the way, if you have to go through FDA approval, right? Usually it's approved for certain indications. And of course, it's possible to do off-label use, but that can be frowned upon in many cases. So there are examples of adult devices being used in, say, the pediatric world. That happens quite a bit, and that's oftentimes not ideal. But again, it has to do with FDA pipeline. You're approved. You have to go through safety and efficacy studies, and then you're approved for a specific indication. If you want another indication, you got go after another approval. So, consumer devices, I think, that don't have to go through approval can certainly be used more broadly. But anything involving regulatory matters, I think that's going to be a limiting factor, and it's there for good reason. So I don't want to overly criticize the FDA. It's they're there for our safety, right?
I mean, yeah, presumably so.
Before that, I think it was a Wild West, and you probably can read about examples of what happened beforehand. So this is sort of a necessary thing.
Yeah. All right. We just received a really interesting question from could you comment on the recent development of brain machine interface by Neuralink? So this is, of course, a brain company or a brain probing company, Elon Musk's company, in terms of flexible polymer and packaging technology. What are the advantages and disadvantages when you compare yours to what?
Yeah, sure. So they are doing, so the Neuralink device is made out of polyimide. And I think that if you ask different people that have worked in these materials, there are sort of advantages to advantages and disadvantages to both. With polyimide, you do have to have, it involves curing. And so it does involve some more specialized, bake steps. With parylene, there are other issues that you have to be careful of when you're processing the material so that you don't sort of cause unintended failures. But in general, I think there's a place for these polyimide devices, a place for these parylene-based devices. A lot of the packaging that they're using right now, at least from what I can glean from the literature that's available, there are some similarities to what we've developed. And I think they have similar challenges as how do you get a large number of connections in a very small space in order to save on the connections that then have to go out of the body. Of course, I think that we are far more cash efficient than Neuralink. They've had lots of investment, a huge team. And so they've had some exciting and interesting results recently in larger animal. So I don't know if that kind of addresses some of the issues there, but I think that in general, polyimide, parylene, there are some similarities. Whether you choose one or the other frequently depends on what material you're most familiar with. That's from a fabrication perspective. But in terms of implementation, my understanding is there's a lot of similarities between the two of them in terms of properties at the end and then what they can do. And so, they could have easily picked, I think, parylene as well.
Okay, so the actual polymer that's being used is hugely important. I think that's what we're hearing here. Okay, thank you for that. One more question. What field would you say presents the largest challenges in biodevices? So I guess to break it down, is it more in the materials? Because of course, everything is moisture absorbent, everything is corrosion intolerant, biofilm, biofouling, etc. So is it the materials? Is it the electrical engineering, the actual circuitry? Is it the biological aspect? What is it?
Yeah, so that's a pretty broad question. So I think it really depends on, how you want to dice that up and what your end goal is. Things that we struggle with, and I actually had a conversation earlier with Kim C about this, is interfaces. Once you insert devices into the body, because we don't fully understand what's going on, if something goes wrong, it's difficult to figure out, what the underlying cause is. So, you could have a failure because of, the construction. Something may be happening to your thin film electrode. You have biofouling. Is there a connector issue? There are a lot of questions out there, and I think really no good answers. There was a review paper that came out, I think 2017, that talks about how difficult and how complex interface engineering is within the neural interfaces space. And so to me, it's not just one field. It's actually a combination of engineering, the surface science, the biology, all of those play a role in that. And I think as we learn more, we'll figure out where, if we can kind of pinpoint one thing, but I actually don't think it's one thing. It's many issues: electrical, mechanical, chemical, biological, all of those play a role.
Wow, I think it really highlights how interdisciplinary and multidisciplinary these problems are, right? So once you get into this device manufacturing world, and especially in the world where it integrates into the body, it becomes so complicated so quickly. Okay, so I think that because it's 5 o'clock, we're going to take one more question. So there's one more that just came in. And, these seminars are not only meant to be the scientific seminars, but we have a lot of young audience members and just people who are in early stages of their careers. And you being so successful, the question is, we'd love to hear your perspective on continuing the translation process from lab into the commercial world. So there are MD, PhD students out there who are clearly choosing not to only practice their clinical skills or their academic research background from the PhD portion of their education. So what would you recommend for this translational type research, and how would one go about that?
Yeah, so I think that being in a biomedical engineering department, I do look towards translation because I do think that if you're building devices that are intended for helping patients down the road, you need to find a way to get it to patients. And so there are a lot of different factors that you have to look at, and I think it depends on where your interest lies. With most faculty, we're not going to leave our day jobs, and so it does take a whole village to commercialize these technologies. And so, so from my perspective, I do what I can in the lab, but ultimately I can only carry things so far, and then it takes others who are, willing to jump in and take the risk and know that, there aren't always answers to all the questions out there, and there's a lot of uncertainty. Take that leap and just push, right? And in many cases, I think in industry, in sort of the entrepreneurial space, my understanding is that failing is actually a good thing, right? Since, and this is sort of contrary to academics, where you don't want to do that, right? You want to get tenure. You don't want to fail in that process. But here, having been through a venture, you learn a lot in that process. And the next time you go about it, you're more prepared to deal with the challenges that you face. And I think running a lab in a way is somewhat like running a small company, but I think there's a lot more certainty than there is in industry, because you don't know. A big factor is ultimately, if you build something, will someone actually buy it? And I think that question is not answered in academic labs early on. It's something that you kind of leave to people to figure out after the fact. And that's a big question if you want to be successful in that space, is who is going to buy it? Just because it looks great in a research paper doesn't mean that it's actually going to help a patient down the road. It doesn't mean that you could actually figure out how to get Medicare to reimburse this medical device. And regulatory and reimbursement, those are two big sometimes non-technical hurdles that you have to pass through, to build successful medical devices. So, I think another thing is that a lot of times when you are doing a PhD, you fall in love with complexity, right? Because that is the thing that is new that you can kind of carve out for yourself. But frequently that complexity is going to create lots of problems for you, especially if you're building a device. Too many issues that could cause you to fail for technical reasons. And so if you look at a lot of the approved devices, they're actually quite simple. And I think it's simple for a reason, because even in that simplicity, there was a lot of very, I think, very good science and engineering that went into making it possible and getting it through regulatory hurdles and so forth. So you got to look at it the long game. This is a long-term process, especially if you're dealing with, say, a class 3 medical device, which many of mine, I think, ultimately will be if they're implanted. And so, there's a lot of work to be done in the lab, and then a lot of work to be done to develop the business case. And all the stars have to align perfectly, right? In some ways, and the funding has to come in, right, to enable you to move to the next step. So maybe it's a long answer to that question.
No, this is such a perfect note to end with. I think that's, again, we're so grateful. I think I'm speaking on behalf of everyone. We're so grateful to not only have such a distinguished Caltech alum come, but to really show us how this whole translational medicine and how to put your devices and fundamental research into practice. It's so great for us to hear. You know better than anyone that Caltech is a very fundamental place. So this is very, very helpful and enlightening. So since we know how to get in touch with you, since you told us where you are, I imagine you might get bombarded with more questions. But for now, in the interest of time, I'm going to just please everyone join me in thanking Dr. Ellis Meng so much, and welcome back to the virtual campus. And I really hope to see you in real life sometime soon.
All right. Thanks so much.
Thank you. All right. Bye-bye.
All right. Bye.